Our presentation is about epigenetic regulation of gene expression in T-cell leukemia. Specifically, we are talking about how tumor suppressor, Ikarus, IKZF1, epigenetically regulates gene expression. Ikarus is a tumor suppressor, and deletion of one copy of Ikarus is associated with about 5% of T-cell ALL and about 11% of early T-cell precursors. In B-ALL, deletion of Ikarus is associated with poor prognosis. So Ikarus is quite important tumor suppressor. So we had a system in which mice who are deficient for Ikarus, they develop T-cell leukemia. Once you reintroduce Ikarus in these leukemia cells, you arrest cells proliferation and you induce differentiation. So what we did actually, we compared epigenetic landscape signature in the leukemia cells and following Ikarus introduction. And what we found is that several unexpected and new function of Ikarus in tumor suppression, which is specifically that Ikarus induces formation of de novo enhancers, also super enhancers, and also activates poised enhancers or activates de novo enhancers. So this is a completely different way of regulation of gene expression that we knew so far. Tumor suppressor is typically a very difficult target. In this case, how we can target, and we showed that a couple of years ago, is that we found that Ikarus protein is inactivated in both B-cell acute lymphoblastic leukemia and T-cell acute lymphoblastic leukemia. And it's inactivated by phosphorylation by oncogenic kinase, casein kinase 2, CK2 kinase. So by targeting CK2 kinase, we can actually restore Ikarus function and reactivate Ikarus to restore its tumor suppressor function. It's mostly leukemia that we know. We know that it plays a role in B-cell ALL, T-cell ALL, most likely AML as well, and pituitary adenoma for sure. There are indications that might play a role in neuroblastoma as well and several other types of cancer, but that's still unsure, it's uncertain. So the next step is with several lines. Certainly we plan to continue our epigenetic studies since now we discover all these new functions. We want to take more into the human primary cells, both B-cell ALL and T-cell ALL. On the other side, we plan to refine our inhibitors of CK2 and to make better drugs for that. And the current existing drugs, I believe that they are heading toward phase 1 trial for hematopoietic malignancies. Well, thank you very much for organizing this meeting. This is an amazing meeting with all experts, world experts in acute lymphoblastic leukemia together. This is quite impressive and it's a pleasure to participate.